I think to think about screening as a paradigm is challenging. I think the way to think about doing that would be to start with overtly promiscuous ligands. So things like staurosporine as a kinase binder or inhibitor, and then build, for sure, almost always more selective degraders, but not supremely selective degraders. Why? Because I think as I was trying to answer the question earlier and sort of generally commenting about the specificity of degraders, once you build a decent sort of, sub-micromolar degrader, the selectivity of these molecules is quite remarkable. And so as a starting point for a screening tool, they're probably not wonderful. At the other end of the sort of degrader spectrum, the so-called molecular glues that have been characterized at the highest level of being able to recognize a beta turn that has a glycine at I+2, which is about 60% of all beta turns, and beta turns are found between beta sheets and helices, and sometimes between helices as well. I think there's tremendous opportunity to build libraries of molecules and screen. As Celgene have disclosed publicly that strategy, it's actually called their CellMod library, so cereblon E3 ligase modulating molecules. And that has been tremendously productive for them as a screening library and a screening tool. It's gotten them into new biology and new target space that you wouldn't find rationally. But it's not making 200,000 molecules. I think they've disclosed that maybe made somewhere between 1,000 and 10,000 molecules. Yeah, absolutely. And I think in my time at Broad, I certainly gained a rich appreciation for the value of big experiments driving big data sets in which you find all sorts of curious and actually often very interesting and powerful things. To do the experiment that you've just described, we certainly have kicked around the idea a number of times. I think the capital cost is extremely high. I love the experiment. I'm just not sure I could convince anyone to bankroll us to do it at this point.
The problem is that not every protein can be degraded by cereblon. And if I have a target, I don't know whether it can be degraded by cereblon or not, but I know that it's cognate E3, it's another protein. So how would you start? Say, okay, should I use a cereblon system, or I need to find another binder for a specific E3?
Yeah, I think it's fair to say that we don't know the limits of any of the three most commonly used E3 ligases. I would urge everyone to divorce themselves from the idea that cereblon or the IAPs or VHL, those are probably the three most significant at this point, have limitations because, for example, VHL has literally only one substrate in a normal biological setting. The IAPs have a handful. Cereblon has 0 that have been characterized to date. Yet those three E3s have been able to degrade an incredible swath of target proteins. And I think the important thing to recognize is that the functional species is not the E3. It's the E3 with a molecule bound to it, and that encodes a true neomorphic function. That species is profoundly different from the E3 alone. I just saw Larry Haman from Celgene, well, I guess now BMS, Celgene's head of chemistry, speak at a conference, and he asserted very strongly that they have yet to find any challenges in using cereblon. They have more experience with cereblon than anyone in the world at this point, and I think that's, again, not N of thousands, but I think it just speaks to this E3 ligase with small molecule bound is a completely different beast from E3 ligase alone.
But if you use the native bonafide E3, you're adding more.
Maybe, except it leaves you with a challenge that if the cognate E3 doesn't have a ligand for it, you're going to have to find it, and the vast majority of E3s are in extremely difficult ligandability classes. Most of them recognize, phosphate residues, very shallow binding grooves. They're, frankly, a hit-finding nightmare from my perspective. Maybe others would feel differently, but I think that's an incredibly difficult path to have. People are doing it nonetheless.
I'm stumbling over a missed connection in my mind. You have these compounds, I'm speaking to the protein degraders, that have high specificity, and then you stop the program due to toxicity. I'm just like brainstorming, is that due to what you brought up with regards to binding a particular ligase for too long, and then there was something that it was supposed to remove from the system didn't get removed, and that's the source of it, or where do you know where it's coming from?
Yes, so toxicity is inescapable, and the desire of fundamentally in drug discovery is on mechanism, on target toxicology. You should find it, and that is what we found there. And it was in a compartment that was not compatible with pushing a molecule forward into people.
In terms of anti-cancer drugs, do you have an approach to estimating the likelihood that tumors will develop resistance, perhaps through utilization of alternative pathways?
This is me. So in this particular program, yes. Okay. And this, at its simplest level, you treat your cell lines with inhibitor for some length of time. Okay. I don't remember exactly what we have for this, but it could be weeks. I've read some people doing this where it literally takes months to develop resistance. Okay. But then after you develop resistance, you look for, for example, proteins that have been upregulated in the system. You say, okay, well maybe that is now, some other pathway that is going on that is compensating for. So that combination trial that combination efficacy study that I showed you came exactly from a study like that. Okay, it was basically there was a compensating metabolic pathway in cells that got upregulated upon treatment with a glutamis inhibitor, and that turned into here's a molecule out there that we know will inhibit that other pathway, so when you combine them, that should knock out that resistance mechanism.
Have you seen discrepancy between the in vitro results and clinical results?
So we just started the trials, don't have the clinical results yet. Okay, but in terms of preclinical animal models, at its very simplest level, just for glutamis inhibition, us and almost everyone else definitely sees less activity in the preclinical models in terms of tumor burden and stuff like that than what you see in vitro. Okay, in terms of some of the combination things we've looked at, across the cell lines, you can have a variety of different mutations. Okay, I don't have exact specifics in my head.
I think actually in that setting, lung adenocarcinomas, which are primarily KEAP1 mutated, that Mick showed, are generally mutually exclusive to EGFR, ALK, or ROS from the other known drivers.
Well, this goes out of my wheelhouse, but thinking back on some other. Going in the other direction, one of the things that we're doing is coming to clinic is that we know in our models, okay, whether in vivo or in vitro, there's some, in this case, there's a particular protein that is over or under expressed in the cell types that we expect to see an effect. Okay, so what we're doing is we're developing an assay to look for that protein level. Okay, so if this is getting where you're going, the reverse part of it would be, what's your CLIA certified assay that actually gives us a marker for a certain cell type that you think is important?
Maybe two questions.
That's all right. You can put an "and" in the middle of it. Yeah.
All right, actually, I'll make it one question, but it'll be to both of you. All right, just to get your big picture thinking. Budgets are limited, resources are limited. What, if you're running the program, and you're obviously running your program, you have a lot of input into your program, what disease do you want to target next and why? And obviously you'll probably be limited to cancer, I would think, Mick, because of where you're at.
We have some neurodegeneration projects as well. We have some neurodegeneration projects.
Yeah, so which one do you want to tackle next and why? Because it's one to say, yeah, we like the pathway, this, that, but the bottom line is, someone's going to have to buy your product for enough money to keep you all in business, right? So what do you want to tackle next and why? What do you want to get out there?
Okay, so there's, I don't think I can tell you because this is an area that the world doesn't know we're interested in. Okay, so from a big picture in terms of indication perspective, I don't think I So something I'm particularly interested in right now, and part of this is because it's at the intersection of some of the neurodegeneration mechanisms and the cancer mechanisms, is actually in the area of cancer supportive care on cancer pain, okay, and chemobrain and things like that, okay. Primarily because this is just not something that is being addressed much. On a personal level, I had someone I loved who died because they literally couldn't take their medications, okay, and it's definitely something that people are more talking to investors, stuff like that, and pitching ideas. People are saying, oh yeah, actually if you could figure that out and even figure out how to run a clinical trial around it, which is one of the big things out there, there actually would be quite a lot of money in there. This is a major unmet medical need. It would definitely trace to patient outcomes so that payers would be willing to pay for it, things like that. I mean, you know, I'm just looking for an excuse to work on something that treats a condition that killed a friend of mine, okay? But you can definitely make the financial arguments for that area. Yeah.
I mean, there's elements of this that resonate for me, probably resonate many people in the audience, but let's just consider your example of glioblastoma for a second, and I'll say something provocative. I think the world has come a long way in terms of treating liquid tumors over the past 3 to 4 decades. And we're not even close to the finish line there, not even close. Solid tumors, most patients who present with solid tumors have ultimately somewhere between a dismal and truly dismal prognosis. Even non-small cell lung cancer patients, you know, average overall survival after an EGFR mutation diagnosed at outset is no more than typically 3 to 5 years. There's plenty of room for improvement in all of these spaces. I like the idea of doing what genetics is already teaching us, and I know this institution here certainly lives and breathes this. Cancer at the level of anatomical presentation is an almost unsolvably large problem. Get into small patient populations and considering what options do these patients have is a great way of approaching what could we do quickly, and it allows one to focus on bringing forward highly differentiated medicines for really high unmet needs, which will always be valuable. I completely reject, at least at the company level, and this does cause me some pain, the idea that we go and solve a problem like prostate cancer, because it should instantly give the question of, well, what type of prostate cancer? Do you mean, you know, castrate-resistant metastatic patients? Or do you mean other earlier stages of disease, and how do we go about that? So getting to small, well-defined patient populations that map to expedient clinical trials where you actually really know who you want to treat, I think is the way in which all organizations can really move the overall needle quite quickly. Yeah, so, you know, look, personally, and it traces a little bit to what Mick was saying. I lost my father to colorectal cancer. He was BRAF mutant colorectal cancer, and BRAF inhibitors are, you know, at least a single agent's not worth a damn in that setting. It looks like the triplet of BRAF, MEK, and EGFR antibodies will be approved, but it's still not a spectacular solution. I think colorectal cancer is one of the examples, like glioblastoma, of a solid tumor, where actually our therapies are not even close to as advanced as others. So there's my personal view, not a corporate view. That's what I'm personally driven by.